Greetings, Chem 1 students. This is the introduction to chromatography. Chromatography is a method for achieving the separation of substances in a mixture. It comes from the Greek word chromos, which means color. What we will be doing is we will be separating different components of mixtures, and we're going to be doing this from colored pencils. Filter paper is a good material in which to use during the chromatography experiment. Okay, so if you read this introduction and then you come on down here and say, in this experiment you will use paper chromatography to separate the dyes of various overhead markers. Okay, so we're going to take filter paper and we're going to take a strip to make our wick also. Okay, I am going to demonstrate before we start the lab on how you will do this. But what we're going to do in our procedure is you're going to draw a line around the center of the filter paper, which is the circular one, the disc one. This will be the point of origin of our overhead markers. You're going to put a small black dot, okay, the size of a print O, at the point of origin. You're going to repeat this with 3 other overhead markers, a yellow one, a blue one, and a green one. I'm going to demonstrate how to apply the wick on this, okay, and how to set it onto the Petri dish in order for the distilled water to come up through the wick and then transfer into your disc of filter paper, and then at the end separate the different colors of the overhead markers. Now, one thing that is important for this is that we have to have a substance that is soluble, in this case water. Well, these overhead markers are all water soluble. If I would have a permanent marker, that wouldn't be water soluble, and this would not work. So I have to have a solvent that will dissolve whatever the mixture is. And in this case, my solvent's going to be distilled water, and it will work perfectly with the filter paper. Okay? What you need to do then is you have to measure distances. You have to measure the DF, which is the distance that the solvent traveled. You also have to measure the DS, the distance that the solute traveled. The reason being is when you come down here, you're going to have to record the DF value right here. You're going to have to record the DS value right here, and you are going to need to have those 2 values to calculate this RF value. The RF value you will read notes on Google Classrooms. So I will post notes on RF values that you need to read through because you need to understand how to calculate that. These notes will tell you how to calculate the RF value. Now, what I can tell you, okay, what I can tell you is if we have primary colors, it's usually only made of 1 single colored compound. If we have secondary colors, it's usually a mixture of 2 compounds. And think about a black. Would that be a mixture? Would it not be? What about green? Mixture of 2 different colors or not? Blue? Yellow? What do you think? Okay? So that's some of the questions here that'll be answered right here. Do the yellow and blue overhead markers appear to be a single colored compound? Okay? Or is it more than 1 compound? So those will be things that you will be answering. All right? So if I would have more than 1 compound, let's say the black one here has more than 1, I would have different RF values for each individual color that the black one had. Okay, so you can expect, I'm not going to lie to you, right here, okay, in this column with the black one, you should have the difference, or the DF, which is the solvent, but you should have many DS colors. And when I mean many, I say, let's say more than one, because you will have more than one color involved with the black marker. Okay. This is Mr. Braun. This lab is titled An Introduction to Chromatography. Chem 1 students, let's investigate.